We presented our first in human trial on CRB401, which is the CAR T-cell protocol in multiple myeloma using bb2121 as the product. Now, this was the updated result with including the expansion cohort as well. So as many as 43 patients were included. What has been used here is a CAR T-cell which is manufactured using a lentivirus against the humanized BCMA, and the co-stimulatory molecule which we use here in addition to CD3 zeta is 41BB, which we think is actually what helps with T-cell expansion and the low incidence of toxicity that we see with this product. We've done a little bit of preclinical work. I think most of us in the myeloma field know now that BCMA is actually a great target, not just for CAR T-cell therapy, but a whole host of other kind of antibody-based strategies. So BCMA as a target was never in doubt. I think the questions which people generally have is, if you have very high circulating levels of BCMA, is that going to impact your treatment or therapeutic strategy? And based on at least preclinical data, we didn't think so. And then the other question I think most people have in this field is that, do you need a certain amount or certain density of the receptor to actually have these therapies work? And preclinically we didn't think that was the case, so we went ahead and started out by doing this product, and we did it in a very classic phase 1 dose escalation trial where we started at 50 million cells and went all the way up to 800 million cells of infusion into patients. We used the classic lymphodepletion. This is what is used in most cellular products to date. That includes Cytoxan and fludarabine given over 3 days, and then 2 days later you give back the leukapheresis genetically modified product. What we found was that the 50,000 dose was really not an effective dose, and anybody and everybody who got more than 150 million cells actually had a response to the CAR T-cell product. What was also very important in this study is, so the 43 patients extremely heavily pre-treated with an average median lines of treatment of about 7 or 8. So basically all of these patients had seen every possible drug which is approved in the context of myeloma today. Some of them in the dose escalation phase had not seen daratumumab, but in the expansion phase we required that everybody had to be exposed to daratumumab as well. So all 5 drugs, carfilzomib, bortezomib, lenalidomide, pomalidomide, and daratumumab, just telling you how relapsed and how refractory this patient population was. The majority of them had had a transplant prior, and a lot of them had had 2 transplants as well. So again, a really heavily pre-treated patient population. What was absolutely striking to me was, going into a cellular product kind of a treatment strategy in myeloma, the concern is always toxicity, but to our surprise and actually our delight, we saw that this was extremely well tolerated. We did see CRS, which stands for cytokine release syndrome, in approximately 60% plus patients. Most of the CRS was grade 1 and 2. We did see a couple patients who had grade 3 CRS, and this was quite easily managed. We used the monoclonal antibody for CRS, which is called tocilizumab. It's an antibody against IL-6 receptor. That was used in all of these patients. Only 9 of the patients ended up getting tocilizumab. Dexamethasone for the treatment of CRS was used in a minority of patients. The other concern most often with cellular therapy is neurotoxicity, and we saw low-grade neurotoxicity in about 30% of patients. Again, it's hard to say that was true neurotoxicity, because when patients have CRS and have a little bit of mental status changes, not quite changes, confusion or not quite on the ball. It's hard to say whether it's CRS versus neurotoxicity. We did have one case of neurotoxicity, which was a grade 4 neurotoxicity, and this was reported at ASH last year in detail. The good news is that patient has recovered and is doing extremely well, and outside of that, we've not had any further neurotoxicity. As far as responses are concerned, these are dynamic as we speak because our expansion cohort was just about finished accruing. So we are still getting responses on that. And that cohort, the response rate, if you look at the data, it looks lower. It's about 53, 54, but that's largely because a lot of the patients have not met their first response date and we've not been able to get all the information. As far as all patients otherwise who have a longer follow-up, the response rate of VGPR or better was seen in 90% plus patients, and this was the first time where we actually demonstrated a progression-free survival in all comers of about 11.8 months, which in this patient population is actually quite remarkable. Importantly, when we looked at patients who had cleared their bone marrow and were MRD negative in the bone marrow and just looked at those 16 patients where we have data on, their median progression-free survival was about 17.8 months. And again, these are unprecedented results which we haven't seen in this relapsed refractory population. So I think to summarize, we have deep responses, we have durable responses, and most importantly, this was well tolerated. I do think we have a lot more work to do with this, and we need to understand should we be better selecting patients. Here we used all comers and end-stage myeloma. Will their lymphocytes, even if genetically modified, work? So there's lots of questions which are out there. Nonetheless, the data is exciting, and we would want to move forward with trying to understand some of these issues.